Hello friends, this video on reproductive health part 13 is brought to you by examfear.com. No more fear from exam. The next popular technique which is often seen is the in vitro fertilization, which is also in short known as IVF. Now, where it helps, what kind of infertile couples it helps? It helps females with damaged fallopian tubes. Now, if the fallopian tube in a female is damaged or it is blocked, now then it is very difficult that the sperm and the egg will be ever able to meet. So, in that case, natural pregnancy is almost impossible. Poor quality of sperms in males. Now, if the male do not have good quality sperms, so that is also another scenario. Now, the question is how it helps. Now, what does the term mean? It's in vitro. The term vitro means glass. That means fertilization takes place in glass. Now, in natural cases, in normal cases where the fertilization takes place in case of human beings, it takes place inside the female body, in the fallopian tube of the females, right? But in IVF, instead of the female body, it takes place outside in the laboratory in a glass vessel, maybe a test tube or any other vessel. That is why it is called in vitro fertilization. And a baby who is born with this technique is often known as a test tube baby. You would have heard about it. It is a very common term being used these days. So that is why the baby is called a test tube baby. Now what happens in IVF is fertilization occurs outside the body. Ovum is collected from the female. So in this case, the female is capable of producing an egg. So ovulation happens in the female, but since the fallopian tube is blocked, so the sperm cannot meet the ovum. So that is the problem in these cases. So the female is able to produce an ovum. So that ovum is taken from the female. Sperms are collected from the male. Now, if the husband has good quality sperms, then it can be collected from the husband itself. Otherwise, the sperm can be collected from some donor. So that is how sperms and ovum are collected as two separate samples. And then these sperm and ovum, they are taken in any glass vessel, maybe a test tube or something, and then fertilization is induced in stimulated conditions in the laboratory. So the fusion between the sperm and ovum, it happens in the laboratory. So this part happens in the laboratory instead of the fallopian tube. Now once fusion happens, what will be formed? Zygote will be formed as a result of fertilization, and then that zygote will start dividing further to form the 2-cell stage, 4-cell stage, 8-cell stage. So that is how the blastomeres will be formed. Now this initial division of the zygote also happens in the laboratory. And then when it has reached the early embryo stage, that time the embryo is transferred into the female's reproductive tract. So that means at that time, if you see, it contains quite a few blastomeres and then it has been directly put into the uterus. So that is how it has been actually implanted into the uterus. So all the thing is done outside, everything is done outside, just that the implantation happens here, so that the growth of or the formation of the embryo to form the baby, that process takes place inside the uterus of the female. So in this case, it is necessary that the uterus of the female is in proper shape, size and is functioning. Now, there can be two stages where this embryo transfer can take place. Now one option is called zygote intrafallopian transfer, which is also known as ZIFT. So this means that the zygote or the early embryo itself is transferred to the fallopian tube. As soon as the zygote is formed, immediately as it divides and form say a 4-cell stage or a 2-cell stage, immediately it is transferred into the fallopian tube. So now if it is at a very early stage, it has to be transferred to the fallopian tube, because under the normal situations, the fertilization happens here in the fallopian tube. Then the early blastomeres are also formed in the fallopian tube. Only when a bigger blastocyst is formed, which has more than 8 blastomeres, only then it enters into the uterus. So if you are transferring the early embryo when it is very early stage, then you have to transfer it to the fallopian tube. So that is called ZIFT. The other option is intrauterine transfer. That means you allow the zygote to grow a little more to form a little more developed embryo in the laboratory, and then you transfer it directly to the uterus. So these are the two options. So one is ZIFT and the other one is intrauterine transfer. So this is how IVF takes place. This is how in vitro fertilization takes place. And the baby which is born later is often known as a test tube baby. That is because the actual fusion between the male gamete and the female gamete had taken place in a test tube. Now, other than artificial insemination and IVF, there are quite a few other techniques also. So, for example, intracytoplasmic sperm injection, which is also called ICSI. So, it is again an in vitro fertilization technique. That means here also the fusion will take place outside the body in the laboratory. But here, what will happen? A sperm injection will be given. So a single sperm is directly injected into an egg. So here also the egg is taken out of the female's body, sperm is taken out of the male's body, and then one single sperm is directly injected into the egg. So when one single sperm is injected, it is sure shot that fertilization will take place, and after that it is again implanted inside the female reproductive tract. So this is called intracytoplasmic sperm injection. Now, how is it different from the in vitro fertilization which we discussed in the last slide? Now, here it just talks about the technique, how the fusion takes place. Now, if just one sperm is injected into the ovum, that is called intracytoplasmic sperm injection. But in the previous slide, what we discussed, we said that the entire sperm sample is put into the test tube, the egg sample is put into the test tube, and then the fertilization was allowed to happen on its own inside the test tube. So that this is different in these two approaches. Another technique is GIFT, which is gamete intrafallopian transfer. Now, in some cases, the gamete itself is transferred from one person to another. Let us suppose if there is a female who cannot produce the female gamete, so the ovulation does not happen, egg is not itself formed inside a woman, and that is why she is not able to reproduce. Now, there can be another woman who would be able to help her. So let us suppose this is the woman who is dreaming of a child, but she is not able to because she is not able to produce an ovum. So now there can be another lady who is capable of producing an ovum, and then that ovum can be transferred from this lady to this lady. So basically she produced the egg is taken out from her body, and then the egg is inserted into her body. Now it has to be ensured that all other parts of her body are functioning fine. For example, her reproductive systems should be good enough to enable fertilization or to enable embryo development. That is, her fallopian tube should be in place and in proper shape and size. Her uterus should be in proper shape and size, so that the further processes can take place smoothly. So this is called gamete intrafallopian transfer. That means gamete is getting transferred from fallopian tube of one female to the fallopian tube of another female. So transfer of ovum from donor to fallopian tube of the mother. So that is known as GIFT. Now you just see how the name is also very strategically put. So she is basically gifting her an ovum. So that is how you can remember it. Another technique is in vivo fertilization, like how we have in vitro. In vitro means something which takes place in the laboratory, in some vessel, that is in vitro. Now, if the fertilization takes place inside the body, that is called in vivo. So, in vivo is just the opposite of in vitro. So, it is to help women who cannot provide a suitable environment for fertilization. Let us suppose this female, she is able to produce egg. She has a proper uterus, but the environment for fertilization is not proper. That is, inside her body maybe there is some issue with the fallopian tube, because of which the fertilization cannot take place inside her body. So in that case, what can be done? Now the transfer of embryo from donor to uterus of the mother. So in that case, what happens? There is another female again who helps her. So this female, what she'll do is, she will produce an egg inside her body. So the male sperm will be inserted inside her body, and then the embryo or the zygote will be formed in her body. Now then the embryo will be taken out from her body and it will be implanted into the uterus of the mother. So only the implantation part and the embryo development part will be taken care by the mother. So rest of the things will be done inside the body of another female. That is why it is called in vivo fertilization. In vitro fertilization, in that what happens is all these processes take place in the laboratory, and then finally the embryo is placed inside the uterus of the mother. In this case, all the processes before the implantation take place inside the body of another female, and then the embryo is just placed inside the uterus of the mother. So that is why the first one is in vitro, because it's happening in the laboratory, and the second one is in vivo because it is happening inside the body of donor female. So here in this case, the fusion occurs inside the reproductive tract of the donor. So that is the difference between in vivo and in vitro. Thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.